Hey, what's going on? Welcome to our introduction to nucleic acids, which are super important in biological systems in terms of their roles in inheritance and protein structure determination. So when we talk about nucleic acids, we're talking about two major molecules. We're talking about DNA and RNA. So in this video, we'll look at the structure of DNA and RNA and how that structure allows them to do the things that they do. We'll look at how biological information is stored and expressed and passed on. And we'll also look at similarities and differences in some of the major biological lineages in terms of how they store their information. So this cartoon diagram shows the two types of nucleic acids, DNA and RNA, and we can see two of the major differences between them right here. First difference that we can see is that RNA is one strand and DNA is two strands. The other major difference that we can see in this particular diagram is the bases. So if we look at the chemical bases in DNA and RNA, there's a slight difference. They both have cytosine, they both have guanine, they both have adenine, but RNA uses uracil, whereas DNA uses thymine. And you can see the difference in the chemical structure. You don't need to know the difference in the chemical structure, but it's definitely a difference between the two types of molecules. The other difference has to do with the sugar. And this diagram isn't actually showing you the sugar that's used, but in RNA, you've got the sugar ribose, and in DNA, you've got the sugar deoxyribose. And that actually gives rise to the difference in their names. RNA, as you can see down at the bottom, is ribonucleic acid because the sugar is ribose, whereas DNA is deoxyribonucleic acid. Let's go and look at the actual chemical structure and see if we can understand what it is that we're looking at. What we have up top on this diagram is a DNA strand, and we know it's DNA because it is double-stranded. And then what we've done is we've taken a section of it and we've just blown it up so that we can look at the chemicals through these chemical diagrams. You can see that we've got chains of nucleotides. And so if we wanted to look at one particular nucleotide, we can just blow this one up here. We've got a phosphate over here, and then we have the sugar, and then we have a nitrogenous base right here. And so that's the overall structure of the nucleotide that we can see here. And we can see they're just chained together. In one strand, they're joined by covalent bonds. Covalent bonds between the phosphate and the sugar. You can see one here, and you can see one here, and you can see one here. And then between the strands, they're held together through hydrogen bonds. So hydrogen bonds, or H bonds as we might call them, are particularly weak bonds, but because there's so many of them in a particular strand of a nucleic acid, it gives a force that holds these two strands together. And you can see that there are certain rules that go along with these bonding between strands. And these rules are as follows. Anytime that we see a guanine on one strand of the molecule, on the other strand, we're going to see attached to it a cytosine. That's always the case. In DNA, we see that anytime that we have an adenine on one strand, on the other strand, we're going to have a thymine. So those are known as the base pairing rules, or the complementarity of the molecule. And so those rules are anywhere you have an adenine on one strand, on the other strand, you're going to have a thymine. Anywhere you have a guanine, on the other strand, you're going to have a cytosine. That's actually really key for DNA's ability to store information, because anytime you have one of those bases on one strand, on the other strand, you're going to have the opposite. Another thing that I'll point out here, it's a little bit beyond the resolution that you need for this course, but I'll still point it out, is that the two strands are upside down compared to each other. You can probably see that if you look at the 5' end that's labeled over here. You can see this phosphate sticking out right here. And if you look at this 5' end down on this side, you can see a phosphate sticking out here. So we would say that this top strand has an orientation of 5' to 3', and this bottom strand has an orientation of 3' to 5'. That's always going to be the case. The geometry of the molecule is such that these two strands are opposite to each other in orientation. We could say they're upside down, but in the cell there is no up and down. So the thing that we use to say this is that they are anti-parallel. And this actually becomes a big deal when we're talking about replication and transcription and how we express the information later on. So it's probably not a bad idea to get a handle on. The only other thing to maybe keep in mind here is that if this was an RNA molecule, we would see one strand. But we still might see base pairing. The RNA molecule can fold over on itself and it can still base pair according to these rules. But of course, in an RNA molecule, adenine would be bonded to uracil and guanine would be bonded to cytosine. So just something to keep in mind there. These two molecules are going to work together in determining the overall flow of biological information inside of a living system. Generally, what we say is that the information in DNA is going to be first copied into RNA, and then the information in RNA is going to be expressed into a protein. This is sometimes called the central dogma, but we can just call it biological information flow if we want. We've got different names for these different processes. The copying of DNA into RNA is known as transcription, and the expression of the information of RNA in protein structure is known as translation. So this is the major way that DNA plays a role in determining the traits and the properties of living things. Because proteins do everything that happens inside of cells, it's the DNA molecule that actually has that structural information and is used to express it throughout the cell. The other thing that DNA serves is it serves as the heritable molecule for life. It's passed on from generation to generation. That process is known as replication, which is the process by which a DNA molecule is copied, and then that copy is passed on to the next generation of cells. We'll look at that briefly here, just so we get an understanding of what this looks like, but the details of replication are sort of beyond the level of understanding that you need for this course. Still, it's kind of cool, so we should talk about it. What happens during replication is that the two strands of the DNA molecule are opened up, and each strand serves as a template for the production of a new strand. And that's all according to those base pairing rules. So you can see in this diagram over here, we have a parent DNA molecule up here, and it is being opened up and replicated. And you can see that we're making two new molecules off of that parent molecule. And everywhere on one of the parent strands, there's an A, we'll put a T. Where there's a G, they'll put a C, etc. So those base pairing rules are really, really important for contributing to the overall heritability of the molecule as well, because each strand can serve as a template by which you can make new strands. Of course, in reality, it doesn't actually look quite this simple. All of the enzymes that are involved in this process all function together as what we would call the replisome. And you can see the different enzymes in the replisome that are all playing different roles here. I'm not going to require you to know the functions of all of the enzymes that you can see here, but the one that I will spotlight is the DNA polymerase, because that is the enzyme that's actually doing the production of the new strand. It's the one that's scanning along the existing strand, finding A's, and then placing T's opposite, G's, and then placing C's opposite. And you can see that there are actually quite a few DNA polymerases that are functioning in this overall replication of this molecule. The other thing that I'll point out is that replication always has to happen making the new strand in the 5' to 3' direction. And you can see that here on this strand that's being made, but you can also see that happening here on these strands that are being made here in tiny pieces that will then actually be stitched together by other enzymes that are operating in this overall structure. Regardless, the end result is that we will have two new molecules from our existing molecule with sequences that are identical to each other. The only other thing that we'll look at in this video is how DNA is organized inside of cellular systems. And so we have a blow-up diagram here. We have the nucleus of a cell that has been enlarged, and if we went into that nucleus, we would find that the DNA is in fact in a series of chromosomes. Now, chromosomes actually only look like this when the cell is dividing. When the cell is not dividing, the DNA is actually much looser inside of the nucleus as what we would call chromatin. But even though it's loose, it still is existing as one particular chromosome molecule inside of the cell. If we look at the difference between chromosomes and genes, we can see that a chromosome actually has many, many genes on it. And a chromosome can actually be quite large. They can be 100 million base pairs in length, whereas genes tend to be a bit smaller, anywhere from 1,000 base pairs, maybe up to 100,000 base pairs, depending upon the particular gene. So we have genes, and those genes are arranged on large pieces of DNA called chromosomes, and those chromosomes are kept inside the nucleus of eukaryotic cells. The other thing that we'll look at in this video is how different biological lineages have organized their biological information. And we can see that in this chart. We've got three major biological lineages: viruses, prokaryotes, and eukaryotes, and we'll compare how their genetic material is organized. Now, of course, remember that viruses are actually not cellular. They are acellular. Many people would argue that they're actually not alive, but they still have genetic material, whereas prokaryotes and eukaryotes are both cellular. We've talked quite a bit about that previous to this video. Viruses will have one chromosome. Prokaryotes will also have one chromosome, whereas eukaryotes will actually have many chromosomes. We'll just put down multiple chromosomes. Viral chromosomes are linear. What we mean by that is if you start on one end of the chromosome and go along, you will run off the other end. Prokaryotic chromosomes, on the other hand, are circular. If you start on one place in the chromosome and go all the way around, you'll get back to where you started from. Of course, it's a big tangle inside of the cell, but it is ultimately a circular structure of the chromosome. Eukaryotic chromosomes are linear as well. So if you start on one end and go all the way to the other end, you will eventually fall off the other side. Viral chromosomes are quite small. Because viruses are not cellular, they infect cells and take them over, they don't need to have a lot of the genes that allow them to live and carry out life functions independently of the cells that they infect. So typical genome sizes for viruses would be anywhere from 10 to the 4, 10 to the 5 base pairs. What we might say is anywhere from 10,000 bases or 10 kilobases up to 100,000 base pairs or 100 kilobases. You wouldn't get much bigger than that. And in fact, most of them won't get much bigger than 10 kilobases. Prokaryotic chromosomes are bigger because prokaryotes have to be independent, self-functioning cells and carry out all of those life processes within that cell. We would find that typical prokaryotic chromosomes would be anywhere from 10 to the 6 or a million base pairs, up to 10 to the 7 base pairs, probably not much larger than that for most prokaryotic cells on the planet, anywhere from about 1,000 kilobases up to maybe 10,000 kilobases. Eukaryotic chromosomes are larger still, but they have a bigger size range as well. They can be anywhere from about 1 million base pairs up to maybe 100 million base pairs, so that'd be 10 to the 9 base pairs. That's a typical range of eukaryotic chromosomes. But of course, you've got multiple chromosomes, so the overall result here is that a typical eukaryotic genome is going to be billions of base pairs. In terms of other notes, I'll just put down that viruses have a tremendous variety of nucleic acids in their chromosomes. So viral chromosomes, for instance, can be RNA or DNA, depending upon the virus. They can be single-stranded or double-stranded. We don't see that in the other lineages. Prokaryotes and eukaryotes, DNA only. DNA is the information storage molecule in every cell on the planet. Prokaryotes do often have additional pieces of DNA as well. They have these small extra chromosomal things called plasmids. They might be 10,000 base pairs in length that they can actually exchange between themselves. We do not see that in eukaryotic cells. And I guess the only other note to keep in mind here is that in eukaryotes, the DNA is found in the nucleus, whereas it's not found in the nucleus in prokaryotes or in viruses. This chart provides a storage comparison of different genomes, and we can see viruses up at the top. You see viruses have maybe 9 genes in the case of HIV, up to maybe as many as 80 genes, whereas once we get to prokaryotes, we start to get into multiple hundreds and thousands of genes. And then when we get to eukaryotes, we see anywhere from multiple thousands up to, tens of thousands of genes, with our human genome having about 21,000 genes in it. Interestingly, wheat, the grain, has 95,000 genes. And the reason for that actually is because its genome is a hexaploid genome. It's got 6 copies of every chromosome. So it's a little bit deceptive here. But generally speaking, as we go from more simple biological systems up through more complex biological systems, we see an increase in the number of genes, which makes sense. In order to be more complex, you're going to need to have more proteins. So that's it for this video. As we move to check your learning, make sure you can do the following things here at the end. Make sure that you can describe the structural and functional differences between DNA and RNA. You should be able to tell me which one you're looking at. And then make sure that you can compare and contrast how the genetic material is organized in different biological systems. If you can do those things, you're doing great. If not, that's okay too. Take a moment, pause the video, and write down any questions that you have so that you can get the answers that you need. Thanks for watching. I really appreciate it. Have a great day.